Hello, everyone. How are you guys? I hope you guys are good and ready to go ahead and get started with this Bio HL past paper. I know it's quite late. I just got home, so sorry, guys. But hopefully you can join me today for this session. Let me know if you guys can hear and see me good. Hopefully you don't hear me double, and we can go ahead and get started. So for today's live, we're going to be doing a Bio HL paper 1A time zone 1 November.
Or
Or
sorry, if the
sorry, if the
audio is
audio is
bad,
bad,
and I
and
can
I
go
can go
ahead and
ahead and
fix it. I
fix
think
it. I
the
think the
audio is
audio
bad.
is
Wait.
bad. Wait 1 second. Is this better? Hello, hi guys. Is the audio okay? Hello, everyone. I hope you guys are good. Yes, it's better. Okay, perfect. Let's go ahead and get started so we don't waste time. So this is going to be a bio HL paper once again. Generally, how I do these, like how I pick the past paper is that I go on our E7 bulletin, which you can just join for free by going on the bio, and I do a poll, and most of you guys said bio, and I remember also from our second account, our past paper account, most of you guys requested HL. So this is once again, this is the November 25 time zone 1 HL paper 1A, okay? Sorry about that writing. I'm going to write that better. But the first question says, what characteristics of the ring seal is a consequence of the viscosity of water? So I've mentioned this multiple times. I've mentioned it multiple times to the students of the crash course, guys. Remember, you need to know not only the properties, the adhesive properties of water, the cohesive properties of water, the evaporative properties of water, the thermal properties of water. Good job. Most of you guys are saying A. Perfect, guys. I'm really, really proud of you. You also need to know, according to the new syllabus, the viscosity and the density of water and how specifically the ring seal and the black-throated bird are able to adapt. And very good. Does anyone know what viscosity means? Good job, guys. Everyone's saying A. I'm very, very proud of you. That is the perfect answer. And I want you to. Oh, someone's asking, is this for real leaked? No, this is an old past paper, like a leaked past paper. Very good. Salva said resistance to flow. Exactly. So you should think of it as resistance to flow. And so what is a streamlined body? Essentially, a streamlined body is talking about how the seal has a very skinny body that protrudes in like a very elongated shape. And so because it has that elongated shape, it's able to pierce through the water better and travel better. Okay, and that's why the answer is A. Good job, guys. Okay, Hershey and Chase used bacteriophages to investigate the chemical nature of genes. The phosphorus in the DNA and the sulfur in the protein of the bacteriophages were radioactively labeled. This is a old syllabus question. It's a repeat question. So if you've been doing past papers, you know exactly how to answer this. I think it's slightly on the harder side just because teachers don't teach this so well. But once you learn it very well, then you're quite good to go. This is the data obtained after the infection and then the centrifugation, of bacterial cells shown in the table. What did Hershey and Chase conclude from their experiment? And we see in the experiment that the concentration of radioactive sulfur was quite high in the supernatant, but low in the pellet. And the radioactive like concentration of phosphorus was really high in the low in the supernatant and high in the pellet. Do we know? Oh, it's a bit blurry. All right, let me zoom in. Is this better? Let me know. We have some A's in the chat. Do you have bio SL papers? Yes, we do also bio SL papers. You can just go on our YouTube. You can go on our second account, Easy7 IB Sciences 2, and I just post past paper working out. Yeah, we do bio SL papers as well. This is just an HL paper because it was requested. I do what you guys request, and this was demanded. Okay, lots of you guys are saying A. Nice. Anyone have any objections in the chat? How do you know this one? I'm going to explain it. Don't worry. This is a bio HL question. So this is only HL syllabus. But it used to be in the old SL syllabus, but now they just moved this to HL. Oh, is this SL? Sorry, guys. The other crash course tutors here. I think this might be HL. But I could double check that for you. Good luck to everyone doing physics tomorrow. Yes, oh my god, I can't imagine. Good luck, guys. So essentially how you should answer this is that you need to understand the difference between the supernatant and the pellet. So a pellet is what's at the bottom of your oh, I forgot what this is called. A flask? No, conical flask. A guys, please remind me. I've had a long day in uni. What is that called? A conical flask, a whoa. Test tube. Thank you very much. Test tube. Thank you. Thank you. Thank you, guys. Okay, a test tube. So essentially, the process of centrifusion is that you put substances in a test tube and you spin it very fast. And because you spin it insanely fast, it separates. And the heavy things go to the bottom, and the light things go to the top. And so your pellet is known as your heavy thing, your section at the bottom that's heavy, and your supernatant is the section at the top. So this is known as your supernatant. Okay, and because bacteria are heavier than viruses, they always end up in the pellet. Okay. And so, what was this point of this Hershey and Chase experiment? Essentially, they had these bacteriophages. Bacteriophages are viruses that infect bacteria. So, my bacteriophage comes here and infects bacteria. How does a virus infect bacteria? It implants its DNA inside the bacteria. So, when there was radioactive phosphorus, we know that the radioactive phosphorus is inside the DNA of the virus, and therefore we know that in order for the bacteriophage to properly inject itself to pass on its genetic information to the bacteriophage, it injected the radioactive phosphorus into the bacteria. And so once the radioactive phosphorus was centrifuged, mixed very, very, very fast, essentially what we found is that there was phosphorus in the pellet inside the bacteria. Now, when it was sulfur, because sulfur is not part of DNA or not part of the genetic information of the cell, essentially it was not injected into the bacteria. This did not happen. And so what ended up happening is that the sulfur stayed in the viruses, did not end up going into the pellet, and stayed in the heavy part, which was filled with bacteria, and stayed at the top. So, what does this show us? It essentially shows us that phosphor is the genetic information of the cell that is passed on. Does that make sense, guys? Determine whether genetic material was DNA or protein. Exactly, very good. And we know sulfur is in protein. You do need to know Chons and CHOP for your syllabus, which is a really good, helpful way to help you memorize that it's carbon, hydrogen, oxygen, and phosphorus in DNA. And, there's nitrogen, oxygen, hydrogen, and carbon in proteins. And there's cysteine, that exception of that amino acid that has essentially that sulfur. Let me know if this makes sense, guys, and I can re explain if it doesn't. Um, Hey, could you explain what supernatant is again, please? Of course I can. So remember, supernatant is essentially just a word to separate the heavy parts that's in the test tube and the light part. And so the heavy part is your pellet, and that's going to be made up of the bacteria. And then you have your supernatant, which is the top, which is made of like your virus because it's lighter. Because when you spin something very, very fast, everything that's heavy is going to go towards the bottom, which is known as your pellet. This real? Do you mean like, is this a real experiment? Yes, it was. Is this a real bio exam? Yes, it is. This is not the predicted paper, guys. Do not worry. We only, at EZ7 in our crash courses, we only focus on um, doing real questions and IB stimulated questions, non AI questions. Okay, chons and chop is for what again? Very good question. So essentially, you should remember chons like this: chons, carbon, hydrogen, oxygen, nitrogen, sulfur is for proteins. This is how I remember the elements in proteins. So that's how I know sulfur and nitrogen. Whenever you see sulfur and nitrogen, you know it's a protein. If you see chop like this, you know it's DNA, which is phosphorus, oxygen, hydrogen, and carbon. Okay? But is this a past paper or a leaked May 26th paper? This is a. Oh, sorry. Okay, now it wasn't clear enough. This is a past paper. Sorry about that. Exactly. Acronyms to remember what elements. Thank you guys for helping each other out. Does anyone have any questions, or are we happy to move on? So, what did Hershey and Chase conclude from their experiment? Viral DNA was found within the bacterial cells. Yes, this is true because the viral DNA was injected, the radioactive phosphorus was injected inside the bacteria and therefore was present inside the bacteria, which we know the bacteria is essentially the pellet, and therefore that's why there was high radioactive phosphorus in the pellet. That's why A is the correct answer. Let's have a read of the other ones just so we're very clear on understanding essentially what the other answer options are saying. Viruses infect bacterial cells with proteins. No, they injected with the genetic material. DNA was mainly outside the bacterial cells. No, it did enter the bacterial cells, and we see that evident. Neither protein nor DNA were chemicals making up genes of the viruses. No, once again, we can cross these out. Very good job, guys. This one I've answered before with you guys. It did show up on the other time zone. Very common IB question, has shown up before. Now, it didn't used to show up, like prior to I think a couple years, and then it did show up, and the crash course tutor put it on her slides, and it actually did show up on the exam. And so be familiar with this. I doubt that this is going to show up on your exam just because it's just it's repeated too often. But I do think you might get like a what is a beta glucose, what is alpha glucose, what is a ribose sugar, a hexose sugar, but not specifically the phospholipid just because now it's too much. Okay, What are we thinking? Is this HL or SL? This is an HL paper. I'm gonna scroll down and I'm gonna ask you to do some active recall. So some of you guys are saying B in the chat. Okay. And can you guys test yourself? Does anyone know what this molecule is here? Because you do need to know what this is. Maybe they're going to show this up on your exam and ask you what this molecule is because you do need to know what it is. Remember, guys, does anyone know what this means? These long chains here. We know, very good job, guys. Everyone's saying for this one, this is a triglyceride. Exactly. You need to be able to recognize that this is a glycerol molecule bonded to three fatty acids. Whenever you see a glycerol molecule bonded to three fatty acid chains, we know that this is a triglyceride. Very good. Now, nice job, Amelia. These lines represent the skeletal structure of fatty acids. So long carbon to carbon to carbon to carbon to carbon to carbon chains. Now, in your paper too, you're generally going to see those long carbon to carbon chains, and actually did show up once in a past paper, or I think twice in a past paper where they did show these long carbon to carbon chains. But generally, you should know for IB in paper 1A, in your multiple choice, you're going to see the fatty acid chains represented by these long skeletal structures. So if this is one, two, three fatty acids, and this therefore means that this one is one and two fatty acids. What do we know, guys, that has two fatty acid tails? And do you guys see something here? Phosphorus. A phosphorus and two fatty acid tails. Phospholipid, and therefore my answer is B. Okay, what do I notice here? I notice that this is a carbohydrate. A carbohydrate, how do I know it's a carbohydrate? Good job, guys. Everyone's saying phospholipids. You guys are eating it up in the chat. Good job, guys. Thank you guys for 1.9k likes, by the way. Really appreciate it. All right. So how do I know that this is a carbohydrate? Well, I can identify that this is a 6 carbon sugar. You should be familiar with the fact that this is a 6 carbon sugar. And you should be familiar with the fact that this is actually glycogen. Does anyone know why this could be glycogen? Let me know in the chat. I'm going to actually leave this one for you to think about this one because I want you to get this is a difficult question. Wait, can you tell us alpha, beta glucose and if those are starch glycogen? Sure, I can. So the difference between alpha and beta glucose, wow, AJ said it in the chat. 1 to 6 glycosidic bonds. Exactly. Good job, AJ. So we know that the bond between a glucose to glucose molecule is glycosidic. Okay, and we know that we need to know that the amylose, which is like this, and we know we need to know amylopectin, which has 1 to 4 bonding. And we know that glycogen has special 1 to 6 bonding. Where do we see that 1 to 6 bonding? 1 to 6 bonding we see here, exactly here. How do I know that this is my 1 to 6 bonding? Well, I can start counting. I can say this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. So I notice that this is the carbon 1 of my one glucose molecule and the carbon 6 of a neighboring glucose molecule. So if they ask you in your IB exam, what type of bonding is this bonding between carbon 1 and carbon 6, or what type of bonding causes branching, then we know for sure it's 1 to 6 bonding. That's the bonding that causes branching. Okay, nice, guys. And how do I know that this is my carbon 1? You always count to the right of your oxygen. I'm going to go ahead and have a read of the chat. Oh, yeah, someone was asking about alpha and beta glucose. So, how do I know the difference between alpha and beta glucose? I'm going to give you a tip that always I used in my exams and I never got the answer wrong. So, remember, you have to know that it's a hexose sugar. This is a really bad hexagon. Sorry, guys. And then you need to know that your carbon 1, which is this one here, is essentially this carbon 1 has a hydroxyl group. And in your beta glucose, your hydroxyl group is at the top, and in your alpha glucose, your hydroxyl at the bottom. How do I recommend that you guys memorize this? Remember that your beta, draw your B's with a large top and a small bottom. And so you know your hydroxyl group in a beta glucose is always at the top. And that's how I remember it. Okay? 1 to 6 bonding, hydrophilic tail, hydrophobic head, very good. Alpha, exactly, very good. What's the difference between this and amylopectin? This doesn't seem very, very branched. This could actually be also amylopectin. You're right. This could be amylopectin. Yeah, but we know amylose is definitely circular. We know that amylopectin, I think amylopectin will have more 1 to 4 bonding than 1 to 6 bonding, if I'm not wrong. This is a good method. Thank you. Of course, I'm glad. Someone's saying save me. If you need help in saving and you like the way I teach and you like the methods I'm giving to help you memorize stuff, I'd really recommend that you guys check out our crash courses. Let me know if you want me to show you a demo. I'm always here to show a demo. Oh, there's an insect. Okay, it says which molecule is paired with a component of cell membrane that allows it to pass through the membrane. And by the way, guys, if you are wondering what this is just 4 amino acids. Okay, nice. Let's have a look here. Someone is saying C. Someone's saying bio is easy, y'all. I mean, good for you that you find bio easy. I think bio was easier in the old syllabus, um, and chem was harder. But now in the new syllabus, bio is deadly. Bio is not easy. If you think about the ecology unit, if you think about all the examples that you have to know for the ecology unit, bio is actually insane. We have C's in the chat. Okay. Anyone have any other idea? Yeah, amylose has 1 to 4 linkages. Amylopectin has 1 to 4 and 1 to 6. Yeah, and glycogen also has 1 to 4 and 1 to 6. So it could be either or. Is this last year's past paper? This is November's past paper. Is my internet bad, guys? Let me know. I feel like I'm cutting off. So the answer is C. Good job, guys. Let's have a breakdown of it. So we know that insulin is a peptide hormone, and you do need to know this for your exam. Unfortunately, sadly, you do need to know it's a peptide hormone. And we know aquaporin. Well, aqua stands for water. And so an aqua pore is just a water pore. And a water pore just lets water go through, and you'll see it in the kidney. So this is where you see HL content being applied, because for you need to know it more for the osmoregulation. So this is incorrect. Glycogen through a channel protein. Okay. We have estradiol through a phospholipid bilayer, and carbon dioxide through a pump protein. Well, pump protein we know requires ATP, it's like active transport, and we know that carbon dioxide doesn't require a protein, guys. My camera's glitching a bit. Oh, I'm so sorry. I don't know how to fix it. I think my internet connection. Thank you for the rose, guys. I really, really appreciate it. So very good, Miguel. Exactly. Carbon dioxide does not require a pump protein. And the reason we know that is because we know through the lungs, you breathe in oxygen and you breathe out um, CO2. And you know that layer has to be very, very thin, and CO2 diffuses inside and outside of the cell along with oxygen. That's why D is incorrect. Now, why is C correct? C is correct because we know estradiol is a what? It's a fatty hormone. It's a fatty hormone, a steroid hormone. Okay, and therefore it's going to be able to diffuse through the phospholipid bilayer very easily. Okay, does that make sense, guys? Now we know okay, very good question. Why is glycogen wrong? Glycogen is a large molecule. Do you think glycogen could go through a cell membrane, or would we have to break it down to glucose? Okay, glycogen probably not. Okay, yeah, and that's I think the key thing that IB exactly. You have to break down glycogen, and that's what happens in the body. You break it down into your glucose, your alpha glucose, and then you're able to properly absorb it. And that's why B is incorrect. Okay, nice. But I'm there with you. Like with my knowledge of IB, I would have been very iffy about B in my exam, but I would have C seems more correct. But I get why B is a bit confusing. You guys are doing so well. It says, which graph shows the effect of pH on the rate of most enzyme catalyzed reaction? This is just a repeat question. It's a nice question to get. I feel like this is one of the good things that my teacher taught me well, that, yeah, that I successfully understood in class. And so I'm going to give you guys a couple seconds, and you guys are answering. You can tell that you guys were taught this very well in class. Everyone's saying A. Yes, guys, be very proud of yourself. So A, the reason why it's A is because we know that enzyme is a protein, and a protein can get denatured. And if you want to do some active recall in the chat, you can tell me what denaturation is. And we know that enzymes or proteins work at a specific optimal pH, and therefore it peaks at one point and decreases at one point, and that's why that's the case. Can anyone tell me what could be replaced with the pH in this specific graph? Because you do need to know exactly why. So why is it not B? The reason why it's not B is because B shows that the as the pH is increasing, the rate of reaction is increasing, and then all of a sudden it decreases. So it shows a point of denaturation. Okay? Now, this one is showing that it peaks at one point and decreases. And that peaking at one point is exactly what we're looking for. Temperature, B is not temperature, B is temperature, good job, exactly. Sorry, I thought you were talking about C. B is temperature, good, because it increases the rate of reaction at one point and then it decreases it down all the way to the bottom. Nice. But what about C, guys? It's another one that you have to know. It's very important. It's actually very common in the exam. It shows up like every other year. If you want, I can. Nice. Bro, Salva's on it. C's concentration. Good job, Momo. Exactly. It's the substrate concentration. And does anyone know what we call this point here? It's a common word to use that's always in your mark scheme. It starts with S, satu, plateau. Nice. Yeah, exactly. It is the plateau. And it's when your enzyme exactly saturated. Saturated. Perfect, guys. This is when your enzymes are saturated. Saturation. Perfect. And then they're just fucking with you. They're trying to pull something. All right, this is an SL question as well. It says the diagram shows a simple respirometer used to measure the rate of respiration. Be very careful, guys. A spirometer is the one you use to measure the lungs, and respirometer is the one used to measure respiration. Soda lime used to absorb carbon dioxide. And actually, this is so nice of the IB to mention that soda lime is used to absorb carbon dioxide because they used to not mention this. They used to expect you to know what the soda lime does. And actually, I think in the M25, they expected you to know it without them telling you. So maybe they realized that students performed really bad because they didn't know the purpose of soda lime. And so they had to mention it in the November. When are you going to go live again? Good question. So if you join our Easy 7 bulletin, if you go in our bio, you can join for free. And I usually do a poll and then I'll let you know when I'm going to go live. How does that work? So this is just something that absorbs CO2. It's not necessarily like a process that works. You have to know that it absorbs CO2. And the reason why they put it is because they want to essentially measure the movement of this drop of liquid based on the respiration. But we know that in the process of respiration, what happens, guys? You uptake O2 inside the organism. It takes O2 in order for it to go cellular respiration, but it releases CO2. And so what ends up happening is the amount of O2 released is equivalent to the amount of CO2 released. So the volume of gas released is equal. You take amount and you release the amount. So you must have soda lime in order for you to absorb the CO2 so we can just measure directly the amount of O2 being taken in. We have some C's in the chat. What unit is this? This is the respiration unit, the cellular respiration unit. This is also an SL question, by the way. So if you're SL, you should be able to answer this. Haven't seen this in IB year 1 yet? Yeah, generally cellular respiration is taught in IB year 2. Okay, we have some C's, B's in the chat. Let's have a read. It says, which way will the drop of liquid move if the organisms are respiring aerobically? So aerobically means that you're using oxygen. The drop of liquid will not move as the volume of oxygen used in respiration equals the volume of carbon dioxide produced. This is true, guys. But what did we just say? We just said that this can't be possible. Why? Who remembers? Because of the soda lime, right? The soda lime is directly absorbing all the CO2, and so it wouldn't be equal. Does that make sense? Exactly, very good. The drop of liquid will move left as oxygen is used by respiring organisms. Yes, right? This is true. It will move left. B is the correct answer. Why is that the case? Well, because as the organism takes in oxygen, this liquid will move and move and move and move because the volume of gas starts decreasing and decreasing, and so the drop of liquid will move to the left. Let's have a look at the other answer option. It says the drop of liquid will move right as carbon dioxide is produced by respiring organism. Carbon dioxide is produced, but it will not move because it's absorbed by the soda lime. The drop of liquid will move right as carbon dioxide is produced and left as the oxygen is consumed. No, we know that that's not the case because we have that soda lime that absorbs the CO2. Okay? Are we happy, guys? Are we happy with this in the chat? I'm just going to close my window really quickly because, okay, perfect. Go ahead and have a read of this one. Uh-oh. We got Krebs cycle. Krebs cycle. This Krebs cycle haunts me every day in medical school and haunts me in IB. Is anyone thinking of studying medicine? Because this will haunt you forever and ever and ever and ever. And by the way, guys, we have free resources on the Krebs cycle. If you go on our website, there's a demo, and we provide you with a free cheat sheet. And I have a cheat sheet, a free cheat sheet, guys. I actually spent so much time making this cheat sheet, so do check it out. It's for free. What are we thinking for this one? Okay, we have some C's in the chat. I want to show you the cheat sheet. Please use the cheat sheet. It was handmade and it took me hours. And it's a sample of the cheat sheet, one of the cheat sheets we show. Where can I find the cheat sheet? I'll show you now. Don't worry. I got you. I got you. So we have a free demo, essentially, that you can just find on the website. So if you go on the crash courses here, okay, and you go down and you press try free demo, you get basically the whole video of explaining respiration very well. And then you can go resources and you have a free resource pack and a respiration sheet. You don't even have to sign up or anything. You can just go and find online. And look, I've created this cheat sheet for you to tell you exactly what you need to say in the mark schemes. And I've made it fun and colorful so that's easy for you to study. Okay. And if you go in the resource pack, there's like sample as well, IB, practice, and model answers. All right, let's go back to the question. When do you open your crash courses for M27? So this will happen next year at like roughly. Oh, when do we open this year's crash courses? April, May, March, maybe March. March next year. March or February. You can sign up. If you go down to the end of the crash course page, there's like a mailing list and we'll email you. Thanks, Dalal. Exactly, March. Crash course student is in the chat. If there's any more crash course student in the chat and you want to let everyone know how you're finding the crash course or how you found the crash course, please do. No, this is going to be like a non-paid review. Be as honest as you want. You can hate on us. Anyway, let's have a read. This is which of the following statements describe part of the Krebs cycle. Reduced NAD is converted back to NAD. Okay, so reduced NAD means what? NADH is converted back to NAD plus. NAD plus. Is this true, guys? Is this true in the Krebs cycle? No, right? We know that it's the opposite that's happening. It's going from NAD plus to NADH. Okay? A 4 carbon citrate binds with acetyl CoA enzyme to 4 a 6 carbon intermediate. Is this one true? Anyone know in the chat? No, it's oxalate. Very good, exactly. If you know your whole revolution, you know that it's exactly. I have a very hard time saying that word, but it's um oxalic acetate. Hopefully, I said that right. Okay, and therefore that's not true. Two decarboxylation reactions occur. Yes, that is true. Why is that the case, guys? Because we know per Krebs cycle, we release CO2. What is a decarboxylation? Well, the whole part of decarboxylation is essentially taking your carbon molecule. This is my carbon, carbon, carbon chain and snapping it off. And we go from, wait, this is not 6 carbons. Sorry, guys. We go from 6 carbons to snapping it off to create a 4 carbon chain. And that's how I know per cycle I release 2 times CO2. And that's why I know that there's 2 decarboxylations that occur. 1 ATP molecule is used in one stage of the cycle. Actually, no, I think it's 2 ATP molecules and 1 GTP, but you don't need to know that for your syllabus. But it's not 1 ATP molecule. Okay, perfect. What occurs in photosynthesis? Can people still join the crash course for M26? Yes, you can. All the recordings are up on the website, and you can watch it in your own time anywhere. You have the exact same recordings. And to compensate for the fact that you guys don't have a live tutor explaining it to you, we have a Discord chat where you essentially are able to add the tutor and message the tutor, and you have access to our emails. You can ask us questions at any point. But yeah, all the recordings are up, and they're divided by subtopics, so you can watch it at your own pace, at your own time, and you have access to all the resources for 2 years. Okay, a lot of you guys are saying D, and you guys are all saying D, Shirley. Nice, guys. D is the correct answer. So, oxidation of NADP occurs in photosystem 1. No. Photolysis occurs in photosystem 1. No. Electron trunking chain synthesize ATP in the outer membrane of the chloroplast. No. We know that these are not true. And we know that these photosystem 1s, their purpose is to emit excited electrons. Ooh, gel electrophoresis. Wow. This was a really, really common question. A really, really common question that showed up all throughout the 2025s, May, November, all of them. So it says samples of DNA can be separated by gel electrophoresis. Which fragments of DNA would travel furthest on the gel? And you should know. Can you zoom in, please? Of course I can. If DNA in the diagram, yeah, of course. Here the diagram, you have your negative electrode, your positive electrode, and you know that DNA is negative, right? All of DNA is negative. Why is it negative? Because we have that negative phosphorus group, that we talked about when we talked about CHOP, right? And so if all of DNA is negative, We know that different sections of the DNA that are cut are going to travel furthest dependent on what? Dependent on their size. Most of you guys are saying A. Good job, guys. Why are we picking A? Because we see that in A, the fragment size is 4. And that is, and along with the 2 1, is much smaller. I'm going to explain why it's not B now. Um, these are the smallest sizes. Okay, the fragment size is smaller in comparison to 10. 10 is a large fragment size. Now, if I ask you if it's going to be harder to push a heavy weight, or if it's going to be harder to push a smaller fragment or a lighter weight, you're going to say it's going to be easier to push a lighter fragment. And that's why a lighter fragment travels further because it's not as heavy and therefore doesn't drag as much. And that's why I can definitely cross out B and C. And we know that it's going to be the fragment with a negative charge. Okay? Because it's going to oppose that fragment of that positive charge. Okay? Perfect. Oh, sorry, it's going to oppose that negative on negative and move towards that positive charge. Does that make sense? Why negative? Well, DNA is negative. How do we know that? DNA has a phosphorus group. Okay, remember CHOP. And so if DNA is negative and I put DNA here next to the negative, what happens when negative and negative are close to each other? Think of a battery or think of a magnet. They repel. And so the negative one is going to try to go as far away, closer to the positive, away from the other negative. Does that make sense? I'm really loving how you guys are helping each other out and helping answer other people. A really good technique is actually, you know, understanding people's questions, understanding why they asked that question, and being able to confidently answer that. So, very good job, guys. Be proud of yourself. The table shows information on the genetic code of 4 different mRNA sequences. Which sequence demonstrates degeneracy in the genetic code? This is new syllabus, and we know degeneracy means what? You have to know this. I gave you a couple seconds. I forgot. Codes for the same amino acids. Hey guys, I just had my first IB exam. Oh my god, how did it go? I hope it went well, Evan. Do we have some D's in the chat? Oh, my iPad closed. D. Different mRNA can code for one amino acid. D. Okay, nice, guys. Good job. D is the correct answer. The reason for that is essentially degeneracy. Someone said it perfectly. Different codons code for the same exact amino acid. Exactly. You have lack of variance. You have that lack of what's that called when you have differences? Variance, no? Essentially, you see here that the same or different, sorry, different codes code for the same exact amino acids, and therefore there's degeneracy, there's that lack of variance. The reason why all of these are incorrect is because you don't have that two essentially here, 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 and here coding for that same amino acid. Does that make sense, guys? And you'll see here, I'm trying to have a look at why B is wrong. Da, 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 da. GUG. Does anyone know why B is wrong? I'm trying to psychoanalyze. CUU, CUU. Ah, yeah, sorry, guys. It's been such a long day. I'm so sorry, guys. It's the same exact codon, and therefore it makes sense that it's coding for the same amino acid. But here, it's different codon coding for different amino acids, for the same amino acids. Sorry about that, guys. Okay? Yeah, exactly. Good job. Perfect. So D is the correct answer for that. Ooh, there's lots of DNA sequences. So 2 DNA sequences are shown. What type of mutations occurring between sequence 1 and sequence 2? Wow. What an annoying question because you just have to like look. This one's fine. This one's fine. 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 I'm just trying to identify where the problem is. Ah, here it is. Okay, I've identified the problem. Okay, what happens when you go from T to A? Is it substitution? Is it polyploidy? Is it insertion or deletion? I think this one's a good one. Exactly. Everyone's saying A. Exactly. So you're substituting thymine for adenine. I'm really liking at the speed at which you're answering this, guys. Be so proud of yourself. Wow. Polyploidy is when you have like multiple limbs. It's just they're trying to trick you. It's IB being confusing. Like multiple fingers. Polyploidy is when you have like extra chromosomes, and I believe I just learned this in med school, guys. Polyploidy. Let me double check my knowledge. Yeah, you have multiple. You have multiple chromosomes, and then you have like multiple fingers and okay. What mode of existence do viruses exhibit? Ooh, a viral one. Focus on viruses, and we know that viruses are essentially all over the new syllabus. Someone said E in the chat. Okay, you guys are saying A. We know that the viruses are parasitic. Why are they parasitic, guys? They're parasitic because they basically feed off the host cell, right? Viruses are not alive, and so they're definitely not predators, and they're definitely not heterotrophs. And what does anyone know what facultative means? Obligate parasite means that they need what? They need a host to survive, exactly, and the host doesn't benefit. Nice, exactly. Facultative means optional. But we know obligate means that there's no choice. Exactly. Very good words. That's why A is the correct answer. Then we're breezing through this past paper. It's been, I think, 36 minutes, and we're already on question 13. Select a microgram source part of a eukaryotic cell. What is the organelle labeled X? This is also on the SL paper. Need a host cell. Wow, you guys are going so fast. Nice. I think this one's obvious. I hate micrograph questions, but this one's quite obvious. Yeah, me too. I hate electron micrographs so freaking much. Can you zoom in a bit? Of course, sorry. Let me zoom in like this and then I'll give you guys a couple seconds and then I'm gonna go down, okay? How are you guys finding this past paper? Is it okay? Let me know. So I'm like grasping how difficult this past paper is. If you want me to try to find a specifically difficult paper 1A. Because a tip for your exam, guys, is that if paper 1A is easy, then your paper 2 is gonna be difficult. And your paper 1B is gonna be difficult. Generally one's easy and one's hard. And so what we can do is I can go in and try to find a really difficult one. For example, for 2023, which was my exam, paper one of chemistry, which was the multiple choice, was so incredibly hard. Some of the questions weren't even chemistry; they were just like logic questions. It was really difficult. But paper two was a breeze. I think I almost got full marks on my paper two. Like I only missed 2 marks. Okay, I'm gonna go down now. What is it? Someone said Golgi apparatus. No. Oh, never mind. A. Okay, nice guys. You guys are saying A. Oh, you guys are saying A. Wait, is my thing loading? Okay, B. Okay, good. You guys are saying B. Smooth endoplasmic reticulum. Nice, guys. It's definitely not A. You should be able to recognize a mitochondria from electron micrograph. How would I recognize it? Via the cristae that it has. And so a mitochondria will look like this and will have a double membrane. That's why this is incorrect. A lysosome will generally look like this, will be very circular, will not have like these folds like this, these large folds. A nucleus will be very dark and it will be huge, okay? And that's why this is incorrect. You like this past paper, it's good. Will you post this solve past paper somewhere? Yes, I can try to post it on YouTube. It might be a bit late, or do check out our second account, easy7ivsciences.com. We're almost at 1,000 followers, guys. I'm trying to hit a goal of hitting 1,000 followers by the end of this week. So if you guys appreciate this live and you appreciate Easy7, hit us a follow on Easy7 IB Sciences 2. It's literally like you spell it Easy7 IB Sciences and then spell the word 2. What's the difference between in the function between rough ER and SER? Very good question. I'm going to answer that in a second. All right, I don't think I missed any questions. If I missed your questions, please let me know, and I'm happy to answer them again. I'm gonna give you guys a couple seconds to answer this while I answer the previous questions. So, what is the difference between the function of RER and SER? So, RER stands for rough ER, and essentially it's the endoplasmic reticulum with ribosomes on the outside. And can anyone tell me what the function of ribosomes are? Hopefully, everyone's able to answer this in the chat. What is the function of ribosomes? If the rough ER has ribosomes, and ribosomes are used to create protein synthesis. Very good job, Daniel. We know that the rough ER's function is for protein synthesis. It's for essentially taking proteins and making sure that they're created and finalized, because you need to know for the HL, guys, pay attention. For HL, you need to know that protein just does not stop at just translation and transcription. It goes a step further where you have modifications via spliceosomes, and you have different modifications of introns and exons and the poly A tail and the methylated cap and the functions of all those. And this is a very big part of the SL syllabus. And this all happens, or the modifications happens within the rough ER, and the rough ER is involved a lot in protein synthesis. Now, the smooth ER does not have those proteins. So it's actually involved in fatty acid, like lipophilic, what's the word? Lipophilic and like fatty cholesterol creation. It's been a long day and I can't remember the word properly in the head, but like lipophilic molecules. Okay? So proteins for rough UR and lipophilic. Nice. You guys are saying B. This is which type of protein uses energy from ATP transfer particles across membranes against a concentration gradient? Against the concentration gradient, we know it has to be active transport. With ATP, we know it has to be active transport. And therefore, you must use a pump protein, just like we've mentioned. A channel protein is facilitated diffusion. An aquaporin is facilitated diffusion. A glycoprotein, does anyone know what a glycoprotein is? You do need to know what it is in the syllabus, and it's actually highlighted a lot in the new syllabus. Who knows the function of a glycoprotein? Very good. It's a cellular receptor for cell recognition. So here I'm doing my phospholipid, and I see that I have a channel protein. This channel protein here is gonna have a glyco, and that's why it's called a glycoprotein. So protein for it being the channel, and glyco for it having this carbohydrate chain attached to it, and that's used as a receptor. Okay. On the phospholipid to be recognized, very careful. It's not on the phospholipid, it's on a channel protein or on a protein. That's why it's called glycoprotein. Very good job, Anushka. Very good job, Amelia. Nice receptor. Very good job, Ivan. Hopefully I said that correctly. So sorry if I butchered your name. The table shows two types of gated ion channels in neurons. How are they gated? Whoa, 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 whoa. I didn't process this question. This is actually no, this is a fine question. I kind of overreacted there. But I just don't like when they mention neonicotinoid acetylcholine receptors. I just do not like when they mention, and especially with the insect example. Yeah, you guys are getting it all correct. Good job, guys. B is the correct answer. Nice. So we know the potassium channels are the channels. We know there's sodium and potassium channels that are lined along the axon. And as soon as a you have your dendrites. These are your dendrites. I'm so sorry. This is horrible dendrites. Your dendrites are very skinny and long. This is not. These are not dendrites. But as soon as a neurotransmitter binds the dendrites, that essentially, and if it reaches that potential, that threshold, that will send an action potential through from the axon Hillcock to the rest of the axon. And the way that electrical impulse is passed is by opening these channels, and these charges that open, these potassium and sodium channels that open, essentially conduct the charge all along the axon. And so we know the potassium channels are definitely voltage, and we know the neonicotinoid and acetylcholine receptors are on that dendrite, and they wait for a neurotransmitter to bind in order for them to start the the action potential. What do you mean fingers? Oh yeah, this looks like fingers. Sorry, these look like fingers, but they're actually dendrites. So your dendrites will actually look like this. So be very careful in case they ask you. Yeah, this is horrible drawing, but this is how your dendrites will look. Okay, nice. Thank you guys for almost 10K likes. I really appreciate you guys. Eukaryotic cells have membrane-bound organelles. What is the property of one of these organelles? The rough endoplasmic reticulum synthesis. Ooh, hopefully you guys were paying attention to the explanation I just said. Rough endoplasmic reticulum synthesizes lipids for use outside the cell. The nucleus has a double membrane to allow vesicle formation for intracellular transport. The mitochondria has enzymes and substrates for the Krebs cycle compartmentalized in the matrix. The Golgi apparatus uses ribosomes to synthesize proteins for export from the cell. Wow. If you guys get this, be very proud of yourself. I think this is just difficult because the phrasing is really weird. Wow. Everyone's getting in the chat. C is the correct answer. Good job, guys. Let's break it down. So, we know that the rough endoplasmic reticulum synthesizes lipids for use outside the cell. Is that true? Is it lipids or what did we just talk about? The rough endoplasmic reticulum. It has ribosomes and therefore it synthesizes proteins. Very good job, Amelia. And therefore this is why A is incorrect. The nucleus has double membrane to allow vesicle formation for intracellular transport. Is that true, guys? Is it for intracellular transport? Nucleus doesn't do vesicles. Very, very good. Exactly. No. We know that the nucleus is as it is, and we know that there's something called nuclear pores, which are holes inside the nuclear membrane, which allow the mRNA molecule to go out and inside. But the nuclear membrane is not for vesicle formation. That's our normal phospholipid membrane. Okay, that's why B is incorrect. The mitochondria has enzymes and substrates for the Krebs cycle compartmentalized the matrix. This is true. We know that the Krebs cycle happens in the matrix of the mitochondria. And then it says the Golgi apparatus uses ribosomes to synthesize proteins. No, no, no. This is the rough ER. But the Golgi apparatus is used for export of materials outside the cell. That's why D is incorrect. Okay? Nice. Someone did a perfect answer, model answer. I want to say just user 349046227. Good job, good job. What is the example of quorum sensing? This is part of the new syllabus, and in the crash courses, we've highlighted exactly the example. So I want to tell you guys something. If you've done the crash course, just by looking at this, without even reading the question and looking at these examples, you would know the answer. I promise you. So hopefully your teacher taught you that example. Good job, guys. You guys are saying C, and that's exactly what we know. What is quorum sensing? Does anyone know what quorum sensing is? Quorum sensing is essentially the process of how bacteria communicate. So bacteria, essentially, when they're a little amount, we have a little amount of bacteria. They barely need to communicate. I mean, they do communicate, and we know that they can through the pili and through conjugation and the movement of plasmids. And this is another part of the syllabus that you need to know. But when there's lots and lots of bacteria, and we know this in this specific example that they mentioned in the syllabus directly, guys, the benefit of the crash course is we tell you the syllabus content, the definition, with the example that's going to show up on your paper 1B, the paper 2, and the paper 1A. And you need to know this specific example for quorum sensing. So you didn't even have to read the answer options. You could have just looked at the example and circled it. And you know that essentially the part of quorum sensing is when there's lots of bacteria, they need to work in unison. They need to work together. And so quorum sensing is all about these bacteria releasing molecules and releasing them only when there's high amount of bacteria so that they can work in unison and work together. And this is an example with this bacterium in the Vibrio fischeri, which actually allows the squid to glow, and it makes the squid glow like this light blue, and it creates a mutualistic relationship where they both benefit because the bacteria gets a home and the squid is scary and it is able to scare away predators. Does anyone know how, like, in what context you need to know this example? I don't know if the crash course student is going to come in clutch here. Does anyone know when you have to know this example? Very good. Very good job. Very good job, Serafima. Oh, is this a Serafima from a crash course? I don't know if this is Serafima, my student, or not. Okay. This is exactly in your inheritance topic that you need to know. This is the example of the new syllabus content you must add. It's part of linked and unlinked genes. Actually, no, this is part of codominance. Yeah, this example is part of codominance, I think, if I'm not incorrect. Oh, yeah, it is Serafima from the crash course. Cute. Nice. Exactly, guys. This one, the chickweed, the mouse ear chickweed, you do need to know that example as well. And this did show up on May in the paper too. So, does anyone know in which example you need to know this Arctic mouse ear chickweed? Hopefully, it's helpful going over these examples because they have shown up exactly those examples. You need to know them. Very good. Nice, exactly. Those are from the crash course as well. The climate change effects, exactly, and the movement of the moose and how that affects the flowers, and if the is it moose that's able to eat the chickweed? Exactly. Good job. And then E. coli is just a very common example that you need to know for like antibiotics and so on. Okay, the image shows a neuron. Which letter shows a dendrite? This showed up a lot also on the November on the SL papers. Hopefully, you guys get this quickly. We have an A in the chat. I like how all the crash course students knew the examples. We got you guys. We got you. Don't play with us. When we tell you the crash course shows every detail, it's literally every detail. Nice. Most of you guys are saying A. Quick question. Circle it and move on. Okay. C is the actual nucleus. Good question, Amelia. That would be the nucleus. So this is called a bipolar neuron. It's a bit outside of your syllabus, but you have a bipolar neuron and a unipolar neuron. A unipolar neuron is the neuron that you know, which looks like this, and it has like the dendrites, and then you have the axon, and then you have the synapse. Bipolar neuron essentially is when you have two axon buddings, but it's the same thing. Okay. And generally, this will be an interneuron. So you do need to know, kind of actually, that in your reflex arc, maybe this will be good review. In your reflex arc, you have what, guys? You have essentially the motor neuron, the interneuron, and then the sensory neuron, right? So you have your sensory neuron, which I'm going to do in blue, and then you have your interneuron, which moves the impulse from the sensory neuron to the motor neuron, so you can move your hand away. And the way generally the interneurons are, they're bipolar, and that's why they have two arms like this, because they must essentially give information in both ways. Okay? Wait, do you need to know how motor neurons and other stuff look like? Um, yeah, kind of, you do. And you need to know how it looks like in the reflex arc. See the cell body, yeah. The nucleus is a cell body. It's the same thing. I think you have to know it works together with interneurons and sensory neurons. Yeah, you do. You need to know also the terms and um, effector and like sensor and so on. The micrograph shows cells in an onion root undergoing mitosis. What is happening in X? Sorry, not C. Unequal cytokinesis by budding. The new syllabus, there's a key highlighting in unequal cytokinesis. I think this will be brought up on your exam. Keep an eye out, guys. Unequal cytokinesis. Does anyone know the example in your syllabus? Hopefully, the crash course students get this. What is the other example in the syllabus? The very common one of unequal cytokinesis. Very good job, Amelia. Oogenesis, perfect. Yeast and oogenesis, good job, Dalal. Exactly, exactly. Good job, Zilsha. Exactly. Good job, Hannah. Nice. Okay, perfect. Assembly of cell membrane and cell wall by vesicles. Contraction of the ring of actin and myosin and proteins. Movement of chromosomes by microtubules. What are we thinking for this one? We have B in the chat. Nice, perfect. So we don't see any unequal cytokinesis here, and we know that happens generally in the two examples that we've mentioned, oogenesis and yeast, and that's why A is incorrect. Contraction of the ring of the actin and myosin proteins, no. Movement of chromosomes by microtubules, no, we're way past that. The chromosomes have been moved, and we're clearly having two separate cells here, okay? Good job, guys. Let me know if I'm going fast. Uh-oh. The water potential of a plant cell is 0.24 kilopascals. If the pressure potential of the cell is 0.46 kilopascals, what is the solute potential of that cell? And they give you the formula. They're actually quite nice. This, I think, is so incredibly deadly, but it was taught in the crash course. And I think we actually did this exact example in the crash course. We have D in the chat. Nice. So the water potential is negative 0.24. Your potential pressure is 0.46, and you're trying to find the solute potential in the cell. And so what do? You just take your 0.46 and you subtract from the other side. So you have negative 0.24 minus 0.46, which will give you the water potential of the cell. Sorry, the solute potential of the cell. And that's why my answer is D. Okay? Nice. Do they always get the formula? Good question. I Mm, I would not trust in that. They're not very clear. This is like the first time this has shown up. Remember, keep in mind, M25 is a new syllabus, and this is the first time that this has shown up. And so I would just memorize the formula because we don't know. Sometimes IB will give you the formula because they're nice, and sometimes they won't. And so you don't want to be that unlucky one who doesn't have that formula. It's quite easy to remember. Just remember solute potential S plus P. Okay, the table shows the binomial naming of the four species. What can be concluded? Will this be on YouTube? Yes, I'm going to try to post it on YouTube. And if it's not going to be on YouTube, it's going to be on our second account, E7 IB Sciences 2. Oh, nice. You guys, the answers are loading. You guys are saying C, 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 B, C, C, C. Okay. C is the correct answer. Well, why is C the correct answer, guys? Well, we know that you have to remember in your syllabus, Katy Perry comes over for grape soda. Kingdom phylum, all of them that in order. And we know as you get closer from kingdom to species, essentially you're getting more and more specific. Okay? And the higher you go up, the closer you are in terms of ancestry. Okay? And so because W and Z have the same genus, we know that they are closer together in ancestry. Because you know species, that means that they are less close or less specific. Okay? But the higher you go up and the higher they have the same classification, we know that the closer they are in relation. And that's why that C is the correct answer. Wait, can you say the Katy Perry thing again? Of course I can. So Katy Perry comes over for grape soda. And actually, the proper one is like great sex, but you have to be like PG. And so they changed it to grape soda. But yeah, that was like the OG old bio one. So Katy Perry comes over for grape soda. You're very welcome. Species related to genus are more closely related than species related to exactly. Very good, Amelia. Damn, Amelia's on it. Genus before species. Exactly. Good job. Okay, good. So, which encircled area shows a clade? This is part of your evolution topic in the HL, and this is a good one to get. I think I would like getting this one in the exam because I'm a very visual person, and so visual questions like this and the phospholipid question would have been very nice for me. Nice. Good job, guys. Everyone's saying D. Yes, D is the correct answer. We know that a clade, essentially, the way I would identify it is that it starts at the same point. And so because this circle is the only one that shows one point and connection, rather than for here, the initial starting points outside of these circles is these two, and therefore this is not a clade. And here as well, you have two separate starting points. This is not a clade, and this is definitely not a clade. And that's why answer is D, because you have that one starting point that buds out to then this one, and therefore this is a clade. The point is called a node, right? Exactly. Very good job. Thank you guys so much for the rose and 14K likes. Are you guys getting tired? Do you want to stop doing the past paper soon, or are you guys okay to continue? Whoa, bore shift. This is a common question to show up. Maybe continue tomorrow? Not tired at all? Okay, I'll wait to see what more people say. We've been going live for an hour now, so you let me know, and we're about halfway through. If you want, we can go faster and finish the whole past paper and go as fast as we can. We have a D in the chat. Let's continue a bit. Okay, let's try to continue a bit. A bit more, maybe we're finished. Okay, continue. Okay, let's try to continue a bit, and then you guys let me know when you're tired, and we can stop and continue tomorrow, or we can do another topic tomorrow. I think the other crash course tutor will be going live tomorrow, so you can guys keep up with that, and then check the bulletin for the timing, or just turn on our notification. So the bore shift is not something new. It's classic. It's common. This is what causes the shift from J to K. What do we notice? We notice that the saturation of hemoglobin with oxygen shifts as the partial pressure of oxygen shifts. And so what causes that shift? The saturation of hemoglobin to shift from J to K. Carbon dioxide concentration to increase or decrease? Nice. Most of you guys are saying D. Good job. D is the correct answer. Now, we know when when carbon dioxide concentration increases, what happens? Does it become acidic or basic? This is very, very important. Hopefully, you guys know this because it's also in the college units all throughout your syllabus. CO2 causes acidic, exactly causes acidic change in your pH, and therefore that causes a lower pH, a decrease in pH, as Amelia is saying in the chat. Good job. And therefore, I can cross out C and I can cross out A, and then, well, actually, wait, look, low key. Let's try to use logic with this. We know a decrease in carbon dioxide concentration would not cause a lower pH. Okay, we know an increase in carbon dioxide concentration would not cause a higher pH. Okay, and then a decrease in carbon concentration could cause a higher pH. Okay, and then you would have to know. Okay, then you would have to know the Bohr shift basically explains how when you have a higher carbon dioxide concentration inside the body, the partial pressure of oxygen increases. So the partial pressure, you can think of it as how well the hemoglobin is essentially attracted to oxygen. And as the carbon dioxide concentration increases, it means you're respiring more. And therefore, you need to increase how well you take O2. And therefore, the partial pressure of oxygen increases as well. And that's why we have that shift. And therefore, D is the correct answer. And this is incorrect. But you could have crossed these out already and you would have had 50-50% chance of guessing. Okay. Acidic with carbonic acid. Exactly. Very good. And that's the example you need to know with the coral reefs. Okay. Nice. Okay. The micrograph shows part of the stem of the common sunflower. In a cross section, what is the name of the function of the structure labeled Z? This is a repeat question. It has shown up before. Loki, maybe the exact same friggin' picture. Partial pressure, oxygen, just how much oxygen there is around hemoglobin. Not necessarily. It's just basically how well it's almost attracted to. Yeah, actually, yeah. You said it correctly. You said it closer to the proper terminology. It is how much oxygen around the hemoglobin. It's kind of like a concentration of oxygen. Can you explain the fetal shift? I can. We do go over that in the crash course. Oh, thanks. Amila's got it. Let me explain it. I feel bad. Sorry. So essentially, the fetal shift, basically, because in when a baby is inside the mommy's stomach, they can't breathe their own air, and so they rely on the oxygen in the mom's blood, and so what ends up happening is that because they rely on the oxygen in mom's blood, they need to have a stronger attraction to the oxygen. So the hemoglobin has higher affinity or higher attraction to that oxygen because they need to steal essentially the blood. They're like parasites from the mom. And so it will shift in a way where there's a higher partial pressure because they require more higher concentrations of oxygen. Hopefully that made sense. Okay? Okay, perfect. All right. So what are you guys thinking for 24? A. Okay, good job. Exactly. A. We know that this is the xylem, and we know that here it's the phloem. And the way I would like to think of it is that the xylem is on the inside where it's larger and so on. Okay, and this is the phloem. Perfect, guys. And we know, sorry, xylem transports water. I would have already crossed out B, and then I would have crossed out C, and then it would have been 50-50% chance, and then it's knowing which one's which. Okay, nice. What is the phloem adapted for? How is the phloem adapted for transport? Thank you guys for 17K likes. I really, really appreciate it. I hope anyone who has an IB exam tomorrow is feeling prepared and ready and rested. And hopefully it's not too late. If it's too late, you should be going to bed. Nice. Most of you guys are saying B. B is the correct answer. We know the phloem has essentially a relationship between companion cells and sieve tube elements, and that's how we aid transport in between the phloem, and we have that movement of that cycle of moving up and down. I think I should go to sleep because I have an exam in 11 hours. Yes, please go to sleep. I wish you the best of luck. Okay? And yeah, I always connected it with the idea of companion cells and then the sieve tube elements, which are hard. And therefore my answer is B. All right, guys. Um, yeah, I think the other ones are kind of Polish A literature paper 2. Okay, good luck. Best of luck. Go girl. Good luck tomorrow. Yes, good luck guys. You guys are going to do amazing. Okay, let's do a couple more questions. This one was a common question that showed up in all 25s. It's a repeat, and they're really, really focusing on this. So pay attention to all that pain reflex arc and that specific terminology. What do we know that the effector is? Best of luck, guys. Um, what topic talks about the reflex thing? Like a response to it. A, so easy, nice, exactly. So effector is the one that's actually causing the effect, the change, not the sensing, and therefore it is the skeletal muscle. Be careful, it's not the gray matter of the spinal cord. You might get that confused because you do need to know what the white matter and the gray matter is according to your new syllabus. Definitely not the pituitary gland, and definitely not the sensory nerve because that's the one that senses things. Okay. Now, yeah, I would just yeah, I think this one's a pretty self-explanatory, but it's also don't get it confused. I think the most difficult one would be the D. Okay. Um, Something for the received signal from a motor neuron, the pain reflex. Exactly, good. Last one on the reflex arc. What topic talks about the reflex arc thing? It's the topic 18 for the crash course. So topic like the human physiology topics. Okay. I'm just trying to see how far we have left. Okay, I think what we should do is end the live here if that's okay with you guys. If a lot of people want to continue, I'm happy to continue just because it's getting really late. But I hope this live was helpful, guys. And we've been going at it for like 1 hour 20 minutes now, and I hope it's been helpful. Does anyone have any questions about anything studying for exams? I'm more than happy to continue this exact past paper tomorrow. It was, thank you so much. You're very, very welcome. So helpful. I'm so glad. Okay, good, good, good. We did 20 questions, and I think we did them like in quite a good chunk amount of time, and we kind of covered lots of different areas of the syllabus. Tomorrow we can continue. Mostly it's going to be ecology, mostly it's going to be examples, it's going to be inheritance. I always love explaining inheritance because I hated inheritance, and so when I get to teach it, I'm like, I got you guys. I'm going to not stop until every single person understands inheritance because I really hated it. Do you have any predictions for topics? I do actually, but my predictions are kind of bad to say them now because generally what I do is I predict them when I'm explaining them, because I predicted based on the syllabus guide. I hate inheritance too. Yes, we can do inheritance with like these inheritance questions tomorrow if that's okay with you guys. Except if Minna's good. Minna might go live, and if she go live, the Minna's the other crash course tutor by the way. If she goes live, she's gonna do. Wait, she told me what she's gonna cover. Wait, 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 wait. Sorry guys, give me a second. If she does go live tomorrow, she's gonna cover. Sorry, guys, wait. She's going to cover neuro and chemical signaling questions. Okay. So if you're interested in that, she's going to go through that. If not, we're going to do this and then cover inheritance and make sure everyone gets inheritance. Is she doing the Discord? I think she might be doing the TikTok live, not the Discord. What time will she go live? Ooh, it depends on when time she gets off the hospital, guys. She's a doctor. I'll let you know. I'll ask her and I'll let you know in the bulletin. I think so. Join the E7 bulletin, and it's for free. You don't have to pay anything. You just join. You go on the on our bio here. I'm gonna show you now. And also, what's nice about the bulletin, guys, is that I upload the past paper working out. There's a drive link on there, and I upload the past paper working out. I'm lost. She's lost. Give me a second. Give me a second. Oh, here you go. If you go here, there's the E7 bulletin, and I post also polls on which past papers I do. So if you go here, I show you that I did a poll and I asked which past paper we should do, and most people voted. 41% voted bio. That's why I did bio. Okay, so I'll do a poll tomorrow if Minna doesn't go live, and I'll ask you if we should continue the past paper. Probably will continue past paper just so we're done with it, and then yeah, I don't know why, but last I checked, the bulletin disappeared for me. Oh no! Can someone else check, please, and let me know if it's also disappeared for you? Hopefully it's still there. I see the bulletin. Okay, good. All right. Maybe you just need to refresh. TikTok's a bit weird sometimes. Don't worry, Seraphim. I can also just share with this past paper with you if you want by phone. It's still there. Okay, good. All right. Yeah, it's not in bio for me either. Oh, really? That's so weird. Ah, if you're under 18, you can't see links. Oh my god, that's the first time I've heard of this. It's like TikTok age. Oh, wow. Damn. You learn something new every day. Okay, I'm gonna post this now on that drive link so you guys have the annotations because I know looking for annotations help. Does anyone have any questions about how to study for exams, or any questions about the crash course, or any questions about uni, about applying to medical school, about IAs if you're year one, about doing the anything? I'm here to like have a chat quickly to kind of cool down after doing this past paper, and I just uploaded the annotations here. Opinions on med school. Wow, do not do it. Yeah, because I no, I'm kidding. If you think you're capable and if you're a person who loves challenges and hard work, then you can do it. I think. Oops, no, I'm just being dramatic. No, guys, I'm being really dramatic. I think I think as a student who was very like I used to be very very confident, and when people tell me, "Oh, don't do medical school, don't do this, it's too difficult," or when I shadowed doctors, they'd be like, "No, no, no, it's too difficult," and I always thought, "Oh, no, it's fine, I can do it, I can do it, I can do it," and but then I actually am in med school. I'm like, whoa, this is crazy. It's actually really, really hard. Med school is really, really hard. It's like beyond your expectations. A lot of people ask me if IB has helped me in med school. I can't even begin to explain how far away, how much more difficult med school is in comparison to IB. It's like just a whole different world, and it makes sense because you're learning how to treat patients and you're learning how to save lives, and so it makes complete sense. But like, I just didn't process that, and I always thought, oh yeah, I'll be fine, I'll be fine, I'll be fine. But yeah, just approach med school with a lot of humbleness and approach med school with a lot of give yourself a lot of grace, basically. Is it too late to run? I feel like you've got it, that honestly. Do you have any friends, people UK who study dentistry? If yes, if you have ever compared how more difficult it is, I think dentistry is hard. I don't think it's as difficult as medicine, in terms of work to life balance. And when I've talked to people who have studied dentistry, I think they're much. I think the content is like difficult because you're in dentistry and you're learning how to treat people's teeth and you're still learning about the human body, but it's not so much a difficult like work to life balance, so you're happier, it's easier in terms of that aspect. Can you explain cyclins? Sure, I can. So cyclins, you mean now? What do you think we should do in the last few days leading up to exams? Very good question. I think all you should be doing leading up to exams is doing past papers. And I've given this tip 10 million times and I'll give it to you guys now. When you're doing past papers, you should not just be doing spamming past papers and writing your mark, your answer, your grade down. What you should be doing is after you do a past paper, you go through and you mark and you write down next to like literally print out your past papers. I was very annoying and I had to print out my past papers on paper because I wanted to stimulate exam conditions. And so I printed out my past papers and I did them by hand. And every single time I got one wrong, I would highlight it in red, and then I would write down, I got this question wrong because I forgot to look at the units. I got this question wrong because I forgot what cyclins are. I forgot this question, and so on, so on, so. And then I would take a picture of that, and then I would save it on the folder on my phone, and it would be like bio wrong, chemistry wrong, math wrong. And then the day before leading up to exams, I literally just spammed insane amount of past papers, did my bio flashcards for bio, and looked at the questions I got wrong. And I went kind of crazy, guys. I did all the past papers from 2013 up until 2010, and I did all time zones as well. And so I had a lot of things that I got wrong, and that's how I ensured that I got full sevens on all my papers. Oh yeah, sleep. Do sleep. I actually cannot be giving that advice because I think in between my bio and chemistry exam, I've slept like it was like 3 days, and I slept like 5 hours in 3 days. It was really really bad. Me too. I like them printed exactly. It's just a different feel when they're printed. I have to print my past papers. I think we're a bit psychotic, but I like it. Um, how much did you get? So in chemistry, I got a six. In bio, oh, six. In chemistry, I got a seven. In bio, I got a seven. In math, I got a six. In psych, I got a seven. And in English, I got a five. One more subject, I forgot. I undersleep two. Chem and bio, HLR. So yeah, they're actually no joke. They're actually really really bad. Yes, sevens exactly. You guys are cool. Yeah, I honestly wouldn't have been able to get sevens without. I was a student. I was a crash course student, and then now I work for Easy Seven. If you guys are interested in also working for Easy Seven after you graduate, let us know. Message us, and you're more than welcome to join. We need more tutors and more IB students that can help and work. It's kind of fun helping other students. Social science. I did psychology. Oh yes, I did French. Thank you. In French, I got a six, or yeah, a six. Let me count my score. Seven in bio. Seven. Honestly, this is the thing. When you get your bio and you get your chem grade and you get your math grade, if you study medicine, that's all you care about. That's all you remember, and then you don't remember anything else. Seven, 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 six, five. Ooh, I got a seven in French. Huh? 7? 7, 7, 6, 5 plus 2. Oh yeah, I got a 7 in French. Oh, that was crazy. How did I not remember that? Any tips for French too? I cannot give you tips. I suck at French. I got an overall 41 points. And so I'm calculating now. If I got a 7 in bio, 7 in chemistry, 7 in psych, 6 in math, 5 in English, 2 points in my minus 41. I got a 7 in French. What math did I do? I did math HL. Hi Joyce. English, yeah, English language and lit. What was your third HL? Oh, no, you're asking someone else. I do English, French, A, and the paper twos are so different. I'm so confused. Yeah, French is just deadly. French is deadly, and you have to like memorize keywords. Any tips in French? Sorry, I just completely like go over your question. I should not be giving tips in French because I suck at French. But tips for French for your writing, you have to memorize. Basically, I only memorize like if it was a journal, or if it was like a diary, and if it was any of those in the writing, I memorized only those, and I would like bet and my lucky chances that it was going to be one of those. And then I would just memorize word for word what I would say. Like I would memorize the introduction, memorize the conclusion, and then I would write 2 sentences about the topic specifically, and then move on. For listening, French actually listening is quite simple, I think, in IB. Reading is where it can get really difficult. So practice your reading. Definitely do a bunch of paper twos for French practice because it's going to be difficult. How did you prefer chem exam? What is the important theory versus calculations? Oh, very good question. I think calculations are more important because once you have to learn theory to get to the calculation part. So focus on centering your teaching around calculations. Think. I think. Yeah, so focus your teaching on calculations because once you master calculations, you've already kind of mastered the theory. Like if you think about the PV equals NRT, do you master that? By knowing to plug in numbers and knowing to plug in sample numbers in order for you to master out the pressure and the volume changes and so on. Also for stoichiometry, that's all calculations. Let's go over the topics. So you have stoichiometry, which is all calculations, and then you have periodicity, which is memorization. Periodicity, heavily common tested topic, and it was tested on a lot in M25, so pay attention. It was actually the most tested topic. Then you have, after periodicity, you have the bonding unit, which is lots of theory actually. Okay, so now you have 2 units on theory. And then you have energetics, which is mostly calculations. Then you have kinetics, which is theory for the SLs, but calculations for HL. Then you have equilibrium, which is heavy calculations for HL. Okay, so it depends if you're HL or SL. In HL, it's definitely calculation based. And then you have acids and bases, which is also calculations based for both HL and SL. And then you have redox, which is kind of calculations. I guess writing half equations is calculations, kind of. And then you have organic, which is just theory. Ooh, someone said for language B, make sure, thanks Amelia for that tip. Make sure to write the answer in reading exactly as it's said or written. That's a really, really good tip, actually. Like literally word for word. Yeah, HL is insanely deadly without the amount of calculations. Do you have any tips for paper two? I don't understand the format. Yeah, French essay convections are very different to English. You're so, so right. Mm, I'm seeing if I miss any questions. Manifesting those grades. Yes, I hope you guys get exactly what you guys are hoping for. And I hope everyone gets in the uni they deserve. I do Spanish B and I'm basically native in it, but easily lose points for not writing, as Rain said. Yeah, that's so annoying. It's really, really annoying how IB you have to say the mark scheme and you have to be very, very specific. Which uni do you go to? I go to Humanitas. Is Med 5 years for you? No, it's a uni in Italy and it's 6 years for us. Okay, I'm just going through the things to make sure I did not miss anything. Okay. Is anyone a non-crash course student? I see lots of crash course students in the comments, which I really appreciate. Is anyone a non-crash course student and want me to show you guys a demo of the crash courses? Let me know. Or if not, we can just end the live here and you guys can go to bed. But essentially, we're actually working on also revamping the dashboard to make it a bit cuter And we're adding a bit. Yes, Mia, let's see. Okay, cute. Thank you, Amelia. Okay, so basically, when you join on the website, this is your dashboard. We have a leaderboard of how many people are studying. Wow, Rosalie's going crazy, and Susanna's also going crazy. And then we have Minna, which is our admin account, and this is how much I've studied. You have your exam timer. We have 13 biology, math, and chemistry. And then you go here. You have your crash course, and once you go to your crash course, my internet's slow. Your favorite science? My favorite science is chemistry, just because I was so bad at chemistry, and I was so happy that I overcame that and was able to get a good grade in chemistry. I got a 7 on all my papers, almost full marks on all my paper 2. So I was very, very proud. My internet sucks badly, guys. What do you think of the cancellation decision? Ooh, for UAE, I think it actually is the best choice. Actually, your exams being cancelled, I know you're in a very, very tough position, and I know that it's valid why it's been cancelled, but I think it's a good, not a good out. What's the word? A good outcome. A good outcome, because I think my predicted, like, for example, I think I got 44 predicted, and I would have loved for that to be the decision, like my final grade, of course. And I worked really, really hard on my IAs, and so I would have loved for that to be part of my final. So I'm assuming that. But also, it might suck for some students, because some students, their teachers suck at teaching them how to do their IA properly, and they don't know the importance of predicted grades. I don't know why my internet's not loading, guys. I'm so sorry. What is happening? What did you do in your EE in? So I always recommend my students to do their EEs in their social science. If you're doing medicine and you're like thinking, let me do a biology EE or a chemistry because that's gonna look better for me, don't waste your time like that. You don't need to do that. Just do the easiest. Do the ones where you can definitely get an A in. And I did psychology, and I got an A in it, and it was actually quite simple to do an A to get an A in it. English is the easiest. No, I generally suck at English, so I don't like I don't recommend to do that, but it's just because I suck at English. There you go. Sorry, guys. I don't know why it took so long. Okay. The dashboard is currently being updated now, which will be very, very cute. There's going to be images here as well. This is the math crash course. For the math crash course, for example, guys, I've created these cheat sheets depending for the students if they've requested in the live session, and I've done these cheat sheets handmade. Nothing's made AI, by the way. And I've created a list of all the sample questions that you can get on the exam, and all the different ways you can input them in the calculator. And I've showed you exactly, basically every single way you can get asked a binomial distribution question, and the same thing for normal distribution. This is kind of the pattern of our crash courses. We compile all the things that you need to know in one place, so you don't have to keep looking for different resources and different things everywhere. I always stress this a lot, that studying for your final IB exams is going to be completely different to how you've been studying for tests and how you've been studying for your mocks. Actually, not mocks, but like your tests and your unit tests, because it's going to be completely different to studying for 2 years of content in one exam. And so you'll see for bio, we've created loads and loads of additional resources that help students. So, for example, this is a syllabus checklist that goes through every single syllabus point that you need to do. Um, and you'll see that because the bio syllabus is structured horribly, 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 we've essentially reordered it in our own way. And if you have a look at this document, you'll see that we've created our own topic 1. And we've shown exactly what topic 1 is and what topic 2 is, because it literally makes no freaking sense to teach students proteins in one topic and then wait till second year to teach them the second part of proteins. This is crazy. Okay, and so we reordered the bio syllabus to make it much, much easier for students to learn. And you'll see that we have lots of cheat sheets. For example, for the menstrual cycle, I have also created the menstrual cycle one. And you'll see here, this is basically all you need to know about the menstrual cycle, the positive and the negative feedback, and all the details here. And you'll see that this is a good example of the menstrual cycle cheat sheet. How much is the crash course? It depends on if you're HL or SL. Yeah, you know exactly what I'm gonna say already. If it's 100, if it's SL, it's 135, and if it's HL, it's 205. Who did physics today? Yes, who did physics, guys? I hope it was okay. Today was the first exam, right? Yes, it was. Yeah, the syllabus is so messed up and randomly organized. It is horrible. It's horrible, horrible, horrible. I don't know why would they do this. Can you show math AA again? Yes, I can. Sure. And so you'll see here we have different types of cheat sheets, and we have the past paper analysis. So essentially, the past paper analysis um, show you all the long answer questions, and they show you exactly the analysis of May 2025 and then 25. And because I do these analysis, I feel like I have a good guess of what can be predicted. And that's why in the crash course videos, as I'm teaching the content, I'm telling you, okay, guys, I have a hint that this is going to show up. And I let you know that this is also all the drawings and the graphs that you need to know. And of course, we have the Anki flashcards. Okay. The chem and physics syllabus are so well organized compared to the bio. It's so funny. Yes, exactly. But also, the bio syllabus used to be organized very well. They just changed it and made it horrible. I don't know why they did that. Yeah, so this is our chemistry one. We'll see here, for example, we have all the ionization energy calculations. For example, for Ka and Kb, these are all the types of questions that you can get asked on Ka and Kb, and I've done it with all the working out, so you know exactly the step by steps, and I've shown on the side the step by steps, so you guys make sure that you are aware of them. For example, there's everything you need to know about buffers. So we do it based on the hard topics that students request in the live. You're more than welcome to request cheat sheets as well, and we're happy to make them for you. And this is everything you need to know on buffers, including the buffer calculations. Keep in mind, guys, that the buffer calculations, you don't have this exact Henderson Hasselbalch equation in your data booklet, and it's not in your syllabus guide, but they are able to ask you about buffer calculations, and they did ask about buffer calculations on the HL question, on the HL exam. And it was an insanely difficult question, and students were expected to know how to calculate it based on equilibrium and the equilibrium calculations. And it was a crazy question, but if you knew the Henderson Hasselbalch equation, because we tell you in the crash course, and we just know it from the old syllabus, then you would have been able to easily answer the buffer questions in the exam. You have summary notes, you have all the different organic chemistry reactions, you have the pH guides, you have the indicators guide, you have so many cheat sheets that are handmade, and you also have the past paper analysis and the common questions that can show up, and multiple choice strategies, because these strategies are very, very important, especially for chemistry. And I think you guys get it, like a hint of exactly the strategies that I teach you guys based on the past papers that I do, because I feel like not only do you learn how to solve questions or which questions are wrong or right, you also learn some of the strategies that I do and I still do in um, medical school. And it's the same exact thing as bio. This is syllabus checklist. You'll see as well that the videos are quite organized nicely. And so you see here, if you go here in your moles, you start off with moles. Here's your video. You can here have a video, the subtopic, and you press on it, or you can watch the full day recording. You have access to the slides, the resource packs. You also have a compiled list of questions, and you'll see, do you have to pay for each subject? Yes, you do. I can give you a discount code if you'd like, but you do have to pay for each subject because you pay for hour of like um, live teaching. I love the recordings. They're literally the best thing. Thank you so much. I really appreciate it. Yeah, I really liked it. Like sometimes the crash course sessions was too long for me when I was a student, so I would just go back and watch the recordings, and I would literally just put on 2 times speed, and I would watch it
Okay,
anywhere,
so we're gonna start
anytime.
off with models of
It was
matter,
so
then we're
fun.
gonna go through the nuclear atom.
And you'll see that we literally go through every single syllabus question. And the way the crash course is organized is that we do content, 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 and then practice question, content, 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 and then practice question. So you're able to know not only how the content's gonna be, you're not you're able to know the content, but you're also able to know how it's going to be applied in your exam. And so here are the resources, which are the slides, and then you have the resource pack, of course, and you have the syllabus checklist that you can cross out to make sure that you fully understood exactly what was in this video. We have flashcards as well that we provide you for the things that you need to know, and we have a discussion section, and then the session, which is just the Zoom link, but the live sessions are over. Okay. Um, yeah, that's about it. And then, of course, for every single topic, you get topical practice questions, and I can show you the ones for bio. So, for example, if you go on day one bio and you go on day one resources, here you have the slides, you have the resource pack, and then you have the topic 2 practice questions. And I'll show you how the topic 2 practice questions are. Essentially, it's every single water question that has shown up until I think 2022. And it's all the MCQs that have been showed up on water with the mark scheme, and then all the response questions on water. And then you have for carbs and lipids, and then the long response of carbs and lipids, and then the proteins. So it's organized in a way that you can watch the video, and then as soon as you finish watching the video, you can go straight to watching, doing the practice questions and so on. Does anyone have any questions? Ooh, someone's talking about that equation p2 squared 2pq plus q squared. Oh, hell no. You do have to know that one. What's that one called? I forgot what that's called. P2. It's on the tip of my tongue and I forgot it. Oh yeah, the Hardy-Weinberg equation. Ugh. Thank you guys so much for joining me for this live. I hope this live was helpful. Please do check out our crash course, check out our resources. Message us on TikTok if you're in need of help. I wish you guys the best of us of luck tomorrow. You guys are gonna do amazing. I hope I see you soon. I'm gonna try to do more past papers to help you guys out before your exam. I hope you guys like this teaching. I hope you like this session and appreciate it. Check out our bulletin. Go through go through that drive with all the past paper annotations. Follow us on TikTok. Turn on our post notification. Turn on all the above. Follow us on YouTube and everything. And I wish you guys the best of luck. And I hope you guys have a restful evening. And you have a very good study session tomorrow. And if you have an exam, I wish you get full marks. Yes, good luck to everyone. Thank you so much. Thank you so much, Amelia, for being so helpful in the chat. I really, really appreciate it. The best IB page in history. Thank you so much. It wouldn't be in a question. No, it wouldn't be in the question. You have to memorize. Yes, good luck. Good night, Sarah. Good night, Emilia. Good night, everyone. Good night, Goofy. Thank you guys so much. And thank you guys for 24K likes. Bye, guys.